All life on Earth is made up of cells, the smallest units of life. Cells carry out all the processes of life, taking in energy and nutrients, growth, responding to stimuli, and reproducing. A cell membrane controls the flow of materials in and out of all cells. Inside the cell membrane lies the cytoplasm, a rich reacting chemical soup of proteins, fats, nucleic acids, carbohydrates, ions, and water. Structures called ribosomes synthesize the proteins found in cell cytoplasms. Structures like ribosomes that carry out specialized functions in cells are called organelles. Cells are divided into two categories based on whether they have one particular organelle, a nucleus. Cells lacking a nucleus are called prokaryotic, Greek for before the nucleus. Cells having a nucleus are called eukaryotic, Greek for true nucleus. The nuclei of eukaryotic cells contains DNA, the genetic material of all living organisms, and several types of proteins, enclosed in a double membrane called the nuclear envelope. No membrane encloses the DNA of prokaryotes, which is usually attached to the cell membrane and concentrated in a region of the cell called the nucleoid. Nucleoids are only distinguishable in that they are somewhat darker than the rest of the cytoplasm. Several other differences exist between prokaryotic and eukaryotic cells. Prokaryotic cells are usually no larger than half the size of the smallest eukaryotic cell and have few, if any, organelles other than ribosomes. Eukaryotic cells, on the other hand, usually have at least 6 or 7 different organelles in their cytoplasm. Another difference is that prokaryotic cells are found only in the form of simple, single-celled organisms like bacteria. Eukaryotic cells are found in more complex single-celled organisms like paramecium and euglena, and all the multicellular organisms on Earth, including humans. Because of their greater complexity, and because they are the building blocks for complex multicellular organisms, in the rest of this program, we'll focus on the structure of eukaryotic cells. Eukaryotic cells can take on a large number of shapes, sizes, and functions. For example, in our own bodies, round, dimpled red blood cells carry oxygen, while long, thin nerve cells that transmit information between the brain and the rest of the body branch out at either end and can measure several feet in length. In the world of single-celled organisms, the slow, constantly changing shape of amoebas allow them to trudge through their environment and capture prey, while their single-celled brethren, the paramecium and euglena, are built for speed. Stiff, non-living coatings called cell walls cover the cell membranes of plants, fungi, and certain protists. The cellulose cell walls of plants and chitin-based cell walls of fungi provide members of these kingdoms with the structural support necessary to stand erect on land. The protective coatings of protists, such as diatoms, which like glass are largely made up of silica, are some of the most beautiful structures in nature. Besides being strong, cell walls are porous, allowing the passage of small molecules such as minerals, amino acids, and sugars to the cell membrane, which actually controls the flow of materials in and out of a cell. Within their cellular membrane, nearly all eukaryotic cells, no matter what their size, shape, or function, contain the following organelles: a nucleus, ribosomes, endoplasmic reticulum, Golgi bodies, mitochondria, vesicles, and vacuoles. Organelles called plastids, of which chloroplasts are a specialized type, are found only in plant cells. Beyond organelles, eukaryotic cells also contain a network of protein fibers that form what is called the cytoskeleton. The cytoskeleton is a network of protein fibers that determines the shape of cells without cell walls, provides for the movement of cells, provides for the structural support and movement of organelles within the cell, and provides the framework for moving and separating chromosomes during cell division. Now let's take a closer look at the organelles and cytoskeleton that make up eukaryotic cells, starting with the nucleus. The nucleus is divided into three major parts: the nuclear envelope, the chromatin, and the nucleolus. The nuclear envelope consists of two bilayered phospholipid membranes, rather than the single bilayer of the cell membrane. The nuclear envelope is riddled with nuclear pores. Nuclear pores are complex structures made up of protein and ribonucleic acid, or RNA, which contain a number of small openings. Water, ions, and small molecules, such as energy-carrying molecules of ATP, can pass freely through nuclear pores. But scientists believe the passage of large molecules, such as proteins and RNA, is regulated by the nuclear pores. The bulk of the nucleus appears granular, with darker and lighter regions, but no obvious structure. This area is made up of a material called chromatin. Chromatin consists of long strands of DNA, parts of which are wound around protein spools. Each complete strand of DNA forms one chromosome. Chromosomes are typically invisible under a light microscope, but when cells prepare to divide, the chromosomes coil up and condense, becoming shorter, thicker, and easily visible under a microscope. The genetic information encoded on the DNA of chromosomes controls processes occurring throughout the cell. In order to exercise this control, information contained on strands of DNA is copied onto molecules of messenger RNA, which pass through the pores of the nuclear envelope into the cytoplasm. Ribosomes in the cytoplasm use the information encoded on the RNA molecules to direct the synthesis of proteins. But communication between the nucleus and the cytoplasm is two-way. Molecules, often proteins, pass from the cytoplasm into the nucleus, providing information on what is happening in the cytoplasm and the extracellular environment. That information is used to regulate the transfer of information from DNA to messenger RNA, and ultimately the production of the proteins necessary to build ribosomes. Though ribosomes carry out their work of synthesizing protein in the cytoplasm, most of the molecular parts out of which they are made are assembled in the nucleolus. The typical ribosome is made of over 40 different proteins and several special kinds of RNA molecules called ribosomal RNA. After the basic components of ribosomes are assembled in the nucleolus, they pass through nuclear pores to the cytoplasm, where they join together to form two subunits, one that holds and reads messenger RNA molecules, and another that holds the protein molecule being assembled. The endoplasmic reticulum, or ER, is a series of interconnected membranous tubes and channels in the cytoplasm, which are usually attached to the outer membrane of the nuclear envelope. Most eukaryotic cells have two forms of endoplasmic reticulum: smooth endoplasmic reticulum, which lacks ribosomes, and rough endoplasmic reticulum, which is studded with ribosomes. Smooth ER has enzymes embedded within it that are the major site for the synthesis of lipids, including the phospholipids that make up the smooth ER, the cell membrane, and the nuclear envelope. In some cells, such as those that make up the gonads of mammals, the ER synthesizes other lipids, such as the steroid hormones testosterone and estrogen. The smooth ER in the liver cells of mammals breaks the carbohydrate glycogen down into glucose molecules that are released into the bloodstream to provide energy for other cells. The ribosomes that cover rough ER synthesize proteins, such as the protein channels and pumps that control the flow of ions and molecules across cellular membranes. In the secretory cells of various glands and other organs, the ribosomes on the rough ER manufacture digestive protein enzymes and protein hormones like insulin. As synthesis of these proteins is completed by ribosomes, they are immediately transported in channels inside the ER to pockets at the end of the ER, where they accumulate and bud off in membrane-bound sacs called vesicles that migrate to the Golgi apparatus. The Golgi apparatus are very similar to smooth ER, but rather than being made out of membranous channels and tubes, Golgi apparatus are composed of stacks of membranous sacs that look like they have been stepped on in the middle, and thus are more narrow in the center and broader or bulging at the ends. The bulges at one end of the Golgi apparatus are the result of vesicles that have budded off from the endoplasmic reticulum and fused onto one end of the Golgi body. The bulges on the other side are created by vessels about ready to bud off from the Golgi body, destined for other parts of the cell. The vesicles from the ER that fuse with the Golgi bodies empty their contents to the Golgi sac, where contents are sorted and processed. The Golgi complex performs three major functions. One function is to separate proteins and lipids received from the ER according to their final destinations. For example, the Golgi complex separates digestive enzymes to be used within the cell from hormones to be secreted to the bloodstream. The second major function of Golgi bodies is to modify molecules. For instance, Golgi bodies modify certain proteins by adding sugars in order to convert them into glycoproteins that can be used as identification markers on the cellular membrane. The final function of Golgi bodies is to package these sorted and modified materials into vessels for transport to their final destination. One type of vesicle formed by Golgi bodies are lysosomes. Lysosomes contain digestive enzymes that break protein, fats, and carbohydrates down into their component parts. Lysosomes perform two major functions: the digestion of food particles and the destruction and digestion of defective or malfunctioning organelles. The food particles which lysosomes help digest can range from individual proteins to complete microorganisms. Some single-celled organisms, such as amoebas, capture their prey through a process called phagocytosis, in which their cell membranes engulf extracellular particles or microorganisms. The section of cell membrane that surrounds the food particle or microorganism then moves into the cytoplasm, forming a food vacuole. Lysosomes recognize these food vacuoles and fuse with them, releasing their cargo of digestive enzymes. These enzymes then digest the food, breaking it down into amino acids, monosaccharides, fatty acids, and other small molecules that diffuse through the membrane of the food vacuole into the cytosol and nourish the cell. Lysosomes digest defective or malfunctioning organelles in a similar manner. Organelles that have outlived their usefulness are enclosed in a membranous vesicle to which lysosomes fuse and release their digestive enzymes. The molecules of the digested organelle diffuse out into the cytosol and are used to nourish the cell and create new organelles. Chloroplasts and mitochondria are energy-producing organelles. Chloroplasts capture energy from sunlight and store it in glucose and other high-energy molecules, while mitochondria extract the energy stored in glucose and other molecules and make it available for cellular activities. Only plants and certain types of photosynthetic protists, such as euglenoids, have chloroplasts. However, all eukaryotic cells have mitochondria. Both chloroplasts and mitochondria are thought to have evolved from bacteria that took up residence within eukaryotic cells long ago. There are a number of pieces of evidence for this. Both chloroplasts and mitochondria have their own DNA and reproduce themselves. Both are about the size of a typical bacteria and, like bacteria, are surrounded by double cellular membranes. Mitochondria are often referred to as the powerhouses of the cell, as they synthesize energy-carrying ATP molecules. It is fortunate for modern organisms that bacteria capable of the aerobic metabolism of glucose evolved, and that some of these bacteria took up residence in eukaryotic cells, as breaking down glucose using oxygen releases 18 to 19 times more energy than the anaerobic method that occurs within the cytosol of eukaryotic cells. The outer membrane of mitochondria can be round, oval, or, as is usual, oblong in shape, with a smooth surface texture. However, the inner membrane always loops back and forth to form deep folds called crista. These folds increase the membrane's surface area and thus the number and rate at which reactions occur. The fluid-filled space between the outer membrane and the inner membrane is called the intermembrane compartment. The area within the inner membrane is called the matrix. Some of the reactions that release the energy stored in glucose molecules occur in the matrix, while the rest are carried out by enzymes attached to the membrane of the cristae. The energy released by these reactions is transferred to ATP molecules that carry the energy out into the cytosol, where it is used by other organelles to power various cellular activities. The process of releasing energy from glucose molecules that occurs in the mitochondria is referred to as cellular respiration. Photosynthesis, the process that combines carbon dioxide and water molecules to create the glucose molecules that fuel cellular respiration, occurs in chloroplasts. There is very little space between the two outer membranes of chloroplasts. The inner membrane encloses a semi-fluid material called the stroma. Within the stroma are interconnected stacks of hollow membranous sacs. The individual sacs are called thylakoids, while a stack of sacs is a granum. Thylakoid membranes contain chlorophyll and other pigment molecules. During photosynthesis, chlorophyll and other pigment molecules capture the energy of sunlight and transfer it to the other molecules in the thylakoid membranes. These molecules, in turn, transfer the energy to ATP and other energy-carrying molecules. The energy-carrying molecules diffuse into the stroma, where their energy is used to build glucose or other sugars from carbon dioxide and water molecules. Completed glucose molecules pass out of the chloroplast into the cytoplasm, where they can be used to build more complex carbohydrates or fuel the process of cellular respiration in mitochondria. Plastids have two membranes and are only found in plant cells. There are two major types of plastids, those that store pigments and those that provide for the long-term storage of lipids and starches. Chloroplasts, which we just looked at, are the most common type of pigment-containing plastids, providing the shades of green nearly universal in the leaves of plants. Other pigment-containing plastids give ripe fruits and certain vegetables their bright yellow, orange, and red colors. Storage plastids are particularly important for biennial and perennial plants that use them to store the products of photosynthesis, usually in the form of starch, during the winter for use the following spring. Potatoes, for example, are made up of masses of cells stuffed with starch-filled plastids that provide the energy for the next year's growth. Most cells contain single membrane sacs called vacuoles. Several types of vacuoles are found in eukaryotic cells. Contractile vacuoles are often found in freshwater protozoans, such as Paramecium. Contractile vacuoles have a network of connecting ducts and a central reservoir connected by a short tube to a pore in the cell membrane. As Paramecium live in fresh water is constantly entering them by osmosis. To counteract this continual inward flow of water, Paramecium collect excess water in the network of ducts that feed the central reservoir. As the reservoir fills, it contracts, squirting the excess water out of a pore in the cellular membrane. Central vacuoles occupy 3/4 or more of the volume of many plant cells. Central vacuoles often store a variety of materials, for example, hazardous wastes that the cell cannot excrete and poisonous substances that deter animals from eating the plant. Central vacuoles also often store sugars, amino acids, and pigments. The colors of many flowers are due to blue and purple pigments stored in central vacuoles. Water entering by osmosis creates the remainder of a central vacuole's volume. Pressure, created by the water filling the central vacuole, pushes against the cell wall, providing the cell with strength and rigidity. The cytoskeleton is a network of protein fibers that determines the shape of cells without cell walls, provides for the movement of cells, provides for the structural support and movement of organelles within the cell, and provides the framework for moving and separating chromosomes during cell division. The three types of protein fibers that make up the cytoskeleton are thick microtubules, thin microfilaments, and medium-sized intermediate filaments. Microtubules are hollow tubes assembled from subunits made out of the protein tubulin. Subunits not forming microtubules are dissolved and scattered throughout the cytoplasm. As subunits are readily available, microtubules can be assembled and disassembled as required by the cell. Subunits are assembled into microtubules by microtubule organizing centers found at various locations within the cell. All eukaryotic cells have a microtubule organizing center near the nucleus, called the centrosome. During cell division, the centrosome produces a football-shaped array of microtubules called the spindle apparatus that separates chromosomes and divides them between the two new daughter cells. In animal cells, a pair of organelles called centrioles are located by or in the centrosome. Centrioles are short, barrel-shaped rings with 9 microtubule triplets forming their outer edge. As a cell prepares to divide, the centrioles are duplicated, and one pair moves to each daughter cell. In cells that have structures called cilia and flagella, the centrioles multiply and move to the surface of the cell, where they become anchored just beneath the plasma membrane, forming basal bodies. 10 pairs of microtubules grow out of each basal body. One pair grows from the center of the basal body, while the other 9 pairs form the outer infrastructure of a cilia or flagellum. A gap, the width of a microtubule, exists between each of the outer microtubule pairs. Numerous motor proteins firmly attached to one pair of microtubules have legs that cross the gap and can walk on the adjacent microtubule pair. The force applied by the motor proteins causes the microtubules to bend. The synchronized movement of large columns of motor proteins attached to microtubule pairs trying to walk up or down adjacent pairs creates the rowing motion of cilia and the wave-like motion of flagellum. Cilia, Latin for eyelash, are short and numerous structures that extend from the cell membrane. Cilia move single-celled organisms such as Paramecium through their environment with a rowing motion. Flagella, meaning whip in Latin, are usually considerably longer than cilia and fewer in number. Flagella move in a wave or whip-like motion. The sperm of most animals and algae rely on flagella for movement, as do a number of single-celled organisms such as euglenoids. Microtubules are also believed to be involved with the movement of organelles, such as vesicles, within the cytoplasm. One hypothesis holds that vesicles and other organelles have receptor proteins to which motor proteins attach and then walk an organelle to its destination via the network of microtubules that extends throughout the cytoplasm. Microfilaments, which are made up of two long chains of globe-shaped actin proteins twisted together, are also responsible for certain types of cellular movement. The muscle cells of animals have thousands of microfilaments running parallel along their length. Thicker filaments, made up of myosin proteins, run next to these. Myosin proteins have legs that allow them to function like motor proteins. The contraction and expansion of muscle cells results from myosin filaments walking in one direction or another along actin microfilaments. Microfilaments are also involved in the flowing movement of cytoplasm in cells, referred to as cytoplasmic streaming, and the formation of cellular extensions called pseudopodia, or false feet, that enable amoebas to move slowly through their environment. Microfilaments play a role in establishing more stable cellular shapes as well. For example, microvilli that extend from the surface of cells of the small intestine in order to increase their surface area are supported by internal structures made up of microfilaments. While both microtubules and microfilaments are involved to a certain extent in the formation and maintenance of a cell's shape, it is largely intermediate filaments, made up of keratin proteins, that form the permanent framework that provides the cells of multicellular organisms with their shape. Intermediate filaments also anchor organelles, like the nucleus, into place, as well as holding certain types of proteins onto the cell membrane and forming the more permanent structures of the cytoskeleton. The shape of cells is critically related to their function. The job of neurons in the brain is to create neural networks that receive, interpret, and act upon information from a wide variety of sources. In order to carry out their role, neurons must branch out in hundreds of directions and make chemical connections with hundreds or thousands of other neurons. So brain neurons have the tangled branching structure with which we are all familiar. Likewise, red blood cells have a mission that involves carrying oxygen to remote reaches of the body through a network of vessels that eventually become so narrow that only one red blood cell can pass through at a time. This function requires that red blood cells be shaped in such a way as to maximize the surface area available for oxygen molecules to bind, while minimizing the chances of the blood cells sticking together and blocking the network of vessels. The rounded edges and concave dimples that increase cell surface area are an elegant solution. These examples of the interrelationship between form and function exist throughout our bodies. Liver cells and those of endocrine glands have a much more fluid form than neurons or red blood cells that allow them to take in and release molecules into the bloodstream. Many of the cells of the respiratory tract secrete mucus and have beating cilia that carry debris and foreign invaders out of the body. While, as we saw earlier, the cells of the small intestine maximize their ability to absorb nutrients by increasing their surface area through large numbers of appendages called villi. It is this incredible diversity in the form and function of cells, the smallest units of life, that makes possible the magnificent living world around us.